All right, so we are recording now. Let's move on into mechanics, skeletal muscle dynamics and mechanics. All right, so I do want to give you a few more terms here. Let's talk about the difference between flexors and extensors. All right, a flexor is muscles that actually move two bones that are attached at a fulcrum, a connection point. And when flexors move the bone, they actually reduce the angle, right? So my biceps are moving my wrists towards my shoulder, and I'm reducing the angle, right? As they contract, I'm moving bone towards my shoulder and reducing that angle. Extensors actually increase the angle. My triceps actually move the bone in the opposite direction, increasing the angle. So the fulcrum is at my elbow. When my triceps contract, they move the bone away from my shoulder, increasing the angle. And these two muscles, the biceps and the triceps, actually have antagonistic effects. And it allows force generation in two opposite directions. So here's a question: how can gastrocnemius muscle contraction result in two different movements? Right? One is actually flexion of the leg, and the other is extension of the foot, right? Both times the gastrocnemius muscle is contracting, but the difference really is the quadriceps. The quadriceps are relaxed when we have the flexion of the leg, and the foot's not planted. So when the gastrocnemius muscle contracts, it picks up the leg, reducing that angle where the fulcrum is at the knee. But in this case, with the extension of the foot, you can see you're increasing the angle of the foot here. Gastrocnemius muscle is contracting, but this time the quadriceps are also contracting, and the foot is planted, so it picks up the heel. Okay, so yes, the gastrocnemius muscle contraction is occurring, but other muscles and whether the foot is planted or not, makes a difference, whether you're getting flexion or extension. All right, here's a little bit of physics. Okay, I know a lot of you haven't taken physics right now, and that's okay. I'm going to try to just kind of walk you through this. In this case, and ask me questions if you're confused by this. In this case, here's the elbow again, and I want you to think about this as the fulcrum. This is where the two bones meet, and this is the pivotal point. It's called the fulcrum, F-U-L-C-R-U-M, fulcrum. This is what's known as a class 3 lever. We talked about class 1 levers before, that teeter-totter where the pivot point or the fulcrum was right in the dead center. In this case, this is a class 3 lever. And we've already talked about this before too, work terms. Remember, work is equal to force times distance. Force times distance. And you can talk about kilograms as a unit of weight, like this 10 kilogram weight, but kilograms can also describe force. So the question is, how much force has to be generated by those biceps in order to hold a 10 kilogram weight out from your body, making sure that this stays in a 90-degree angle. All right, so the work terms are this. The biceps are attached to your bone, your forearm, from the fulcrum to about 5 centimeters. And then another 30 centimeters out is where you're holding the 10 kilogram weight. So the work term is this, 10 kilograms times 35 centimeters, right? Now, the bicep is attached here at this 5 centimeter mark. How much force, right, do we have to generate in order to keep that 10 kilogram weight out in this 90-degree angle? And if you do a little bit of algebra, high school algebra here really, x is going to be, that's what we're looking for. Multiply that by 5 centimeters, right? And we're going to equal that to 35 centimeters and that 10 kilogram weight. If we just solve for x, that means that we have to generate 70 kilograms of force in those biceps to hold a 10 kilogram weight out 30 centimeters from the site of the muscle attachment. Okay? It's kind of interesting. It sounds like that would be a disadvantage, right? It'd be more of an advantage if our muscle, our bicep, was actually attached at our shoulder and our wrists, right? Then we wouldn't have to generate that much force in order to pick up that weight. But you can imagine if our biceps were attached at our shoulder and our wrist, that would be super awkward, right? We wouldn't have much flexibility. Yes, question?
Okay, I'm confused with the math. Is it divided by 5 centimeters?
Yeah, so it would be this, right? If 10 kilograms, and we're multiplying that by 35 centimeters, right?
350.
Yep, 350. And then we've got x is how much force needs to be generated by the biceps times 5 centimeters. So we solve for x, we're just going to divide this by 5 centimeters, right? That's going to cancel out, and that's going to be 350 divided by 5, which would be 70. Yep. Yep, sure. All right. So what is the mechanical advantage? Why have we evolved this way? Well, this is where the advantage comes into play. You can see here that just a 1 centimeter movement close to the fulcrum here translates to a 7 centimeter movement at the hand. And you get similar gains in velocity too. So velocity, just, a 1 unit of velocity here close to the fulcrum is actually going to translate to 7 times the hand velocity. So that's important. Here's the take-home message here. This is the biological advantage. Number 1, a small degree of shortening produces large movements of the hand. And the speed of hand movement is significantly increased relative to the initial shortening velocity. So you get similar gains in velocity. This gives us a biological advantage. Yeah, I would say that if you're talking about the pivotal point, if you only had to move, well, this is what's happening. You only move this part of your arm 1 inch, but you're actually getting 7 inches at your hand. It's, right? 7 inches. And the speed is the same. You only have to have a certain shortening velocity here at the fulcrum. That translates to 7 times the velocity at your hand. So it gives you a biological advantage because of those gains in both distance and velocity. Yep. All right, so Duchenne's muscular dystrophy actually weakens these lever system relationships used to help bone stand up. So basically when you're talking about Duchenne's muscular dystrophy, you're talking about a protein that doesn't get to the membrane. It's called dystrophin. And that actually really weakens the muscle. And it weakens the muscles that are responsible for posture. It's a very signature symptom of Duchenne's. And there are some heart issues as well with this type of muscular dystrophy. It alters that lever system. All right, so let's move away from skeletal muscle and talk about smooth muscle. There's a few things that I need you to know. Smooth muscle surrounds, it actually kind of wraps around different tubes in your body. Whether we're talking about the vasculature, your blood vessels, or structures like your small intestines. And when this smooth muscle contracts, it actually decreases the radius of these tubes. And then when it relaxes, it increases the radius of these tubes. So with smooth muscles, sometimes there are types of smooth muscles that have slow, regular contractions. One really good example of that is in your small intestines. Those smooth muscles that surround the small intestines can be rhythmic, right, to help with motility and movement of food down the tube. And it's kind of interesting, actually. Let's see here. They can have these interesting contractions. We're going to be talking with cardiac muscle of a particular channel that causes an eroding depolarization, right? These are called pacemakers. So with pacemakers, they just reach threshold all on their own. These are calcium spikes that cause contraction, relaxation, contraction, relaxation. So we'll talk about these pacemaker cells. They are in your gut as well, right? Helps to give you those rhythmic contractions. Other types of smooth muscle cells, like in your vasculature, can cause prolonged contractions. Vasoconstriction is one of them. When you have a surge of that sympathetic tone, you get scared or frightened. A lot of times your vasculature in the periphery to your fingers are going to contract. So you're not delivering as much blood to your fingers. You're actually redirecting a lot of the blood back to your core organs, like your heart and your brain, in case you have to run or be strategic about fighting. So those are prolonged contractions that can cause vasoconstriction. And smooth muscle contributes to many different systems. We're going to be talking about this with the GI tract, obviously, kidney. Now, there's some key differences with skeletal muscle. They're not organized in sarcomeres. Remember, these aren't striated muscle. They do not have T-tubules. And they have very small, minimal sarcoplasmic reticulum. With that, a lot of calcium comes from the extracellular space as well as internal stores. Now, we talked about gap junctions with epithelial cells. These cells, some of them called single unit smooth muscle, have gap junctions. So that means the calcium goes from cell to cell to cell, and all of those cells contract as one single unit, called a single unit smooth muscle cell, smooth muscle tissue. They also contract in all dimensions. Let me go to the next slide. I can show you what that means. You can see here when it's relaxed, it's kind of organized this way, but then when they contract, remember, smooth muscle does have actin and myosin, and calcium is important. When the actin and myosin can actually interact with each other, it contracts in all directions. A lot of times around tubes, it'll contract and cause constriction, decreasing the radius. So smooth muscle do have actin and myosin, those thick and thin filaments. Again, remember the thick filaments are myosin, the thin filaments are actin. And this causes muscle contraction. But they don't have troponin, they don't have tropomyosin, they don't have T-tubules, and they're not organized in sarcomeres. They don't have striations. All right, so I would star this particular diagram. This is giving you all the information that you really need about smooth muscle contraction. It does differ from skeletal muscle. So what you see here, the similarities between all three muscle types, skeletal, cardiac, and smooth muscle, is they all depend on an increase in intracellular calcium. So once calcium floods into the intracellular space, it activates an enzyme called calmodulin. This is a kinase. So it activates calmodulin. Calmodulin, the activated calmodulin, phosphorylates myosin light chain kinase. Now, if you go back and you think about myosin, the anatomy of myosin, it had a tail region and a head region. The myosin light chains are actually on the neck region. So it phosphorylates the myosin light chains that are on the neck. And once they're activated, these are active calcium calmodulin myosin light chain kinase. Once it's activated, it actually activates the cross bridge. So you get a 90-degree angle of the myosin head. It can now interact with actin, and you get crossbridge cycling. You already know about crossbridge cycling. You actually have ADP and inorganic phosphate. When those actually dissociate, remember you get the power stroke. ATP is required to dissociate actin from myosin, and the hydrolysis of ATP re-energizes the bridge. So you get cross-bridge cycling and shortening in all directions, causing vasoconstriction, and it just keeps going as long as calcium is around. Once calcium is either removed across the plasma membrane or re-sequestered into those internal stores, then the myosin light chain phosphatase. We talked about kinases before. Kinases are enzymes that phosphorylate stuff. Phosphatases dephosphorylate. They take that phosphate group off. So then the myosin light chain phosphatases dephosphorylate, and an unphosphorylated myosin is held near the thick filament and cannot interact with actin. Okay, so if you need a little bit more text, you can always read about this in your textbook, but these are always nice flowcharts also to really compare and contrast what's happening with smooth muscle and skeletal muscle. So this is something that I'll just have you kind of read on your own. It's a review of what I just talked about, but it's a nice flow chart to really compare and contrast the two sequence of events. And remember, calcium is important for both, and myosin and actin interactions are also important for both. All right, so going back, what are some of the other influences, the inputs that influence muscle contraction? We talked about the first one. You have some spontaneous rhythmic contractions. Neurotransmitters we also talked about could be important with that fight or flight response. You can get sustained contraction. Also, hormones are important. We're going to get to kidney, and we're going to be talking about vasopressin, which you can imagine causes contraction, or angiotensin, again, another hormone that actually causes contraction. You don't need to memorize that. Just know that hormones can play a part in smooth muscle contraction as well. Also, local changes, right? With a lack of oxygen, you can imagine your tissue, when it is really deprived of oxygen, it will send local paracrine signaling molecules to cause vasodilation to actually help with oxygen delivery. The opposite is if there's a lot of oxygen, then it's going to cause vasoconstriction. So acidity, oxygen osmolarity, different ion concentrations can all contribute. These are local changes within the environment of smooth muscle cells. And then finally, stretch. Sometimes there are stretch receptors, mechanically gated, that cause calcium to flood in. So imagine a surge of blood going into a tube, right, a blood vessel. That stretch can activate stretch receptors and calcium channels that cause a compensatory constriction by smooth muscle to try to keep the flow within homeostatic range. So all of these inputs are important. All right, so let's make sure that I haven't forgotten anything. I feel like we covered that pretty well. Okay, the difference between single unit and multi-unit smooth muscle, again, as I talked about before, with single unit, A up at the top here, let me blow that up. You can actually see these have gap junctions. This allows for calcium, once it floods into the intracellular space, to easily move from cell to cell to cell, so that all of these cells contract as one unit. When this motor neuron is activated, all of these smooth muscle cells contract as one single unit. Helps to coordinate the contraction. Multi-unit is the opposite. It actually doesn't have gap junctions. So only this motor unit is going to contract. Not all of the other motor units will. So this is a multi-unit smooth muscle organization. All right, that's it for smooth muscle. It's a lot of information, but this is probably the one that I really want you to study. This is what's happening within the smooth muscle cell. All right. And then finally, let's start talking about cardiac muscle. All right, so here's two other terms, the difference between myogenic and neurogenic. Myogenic and neurogenic. So I don't know how many of you guys play video games. There used to be a video game, I can't remember the name of it because I'm not a gamer, where, and somebody's probably going to tell me in just a second, where basically the two fighters, they're warriors, one actually reaches into the chest cavity of the other one, pulls out their heart while it's still beating, and shows it to him before he keels over and dies. Do you guys remember that? That seems pretty violent, right? I'm going to have to look it up. I'll tell you on Wednesday. But my question to you is, if I actually pull out the heart and put it into some kind of box that has enough oxygen and nutrients, will that heart continue to beat? What do you think?
Oh, I was going to say that video game is Mortal Kombat.
Yes, Mortal Kombat. Yes, that's it. Mortal Kombat. Thank you. All right, Mortal Kombat. Okay, so if I put this into a box and I allow it to have enough oxygen and nutrients, do you think it will continue to beat? What do you think? If I can keep all those cells alive, will it continue to beat? Yes? How about you talk to your neighbors for a second? Will it continue to beat? This was actually a Grey's Anatomy episode, actually. It was called Heart in a Box. Yeah. 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 A whole heart, yeah, whole heart. without any neurons, without any input from hormones, will it continue to beat? What was that? Thank you for that. Mortal Kombat. I couldn't think. Yes. All right, what do you think? Who wants to take a guess? Yes. That is the correct answer. Yes. So, what that means is we're going to be talking about the SA node. The SA node are the pacemaker cells of the heart. They don't have a nice, steady, resting membrane potential. They have an eroding membrane potential that always reaches threshold and causes action potentials and contraction. So what that means is the cardiac pacing is within the muscle itself. That's why it's called myo meaning muscle, genic meaning origin. So the heartbeat is initiated within the muscle itself. So it contracts spontaneously. There's a spontaneous contraction. And because of gap junctions, the action potentials, and all of the depolarization wave goes from cell to cell to cell, right, because of these gap junctions. So you get a coordinated heartbeat. That's really important in cardiac muscle, unlike skeletal muscle. Skeletal muscles don't have gap junctions. So you want that depolarization wave to go from cell to cell to cell so that you get a coordinated heartbeat, so you can propel blood out. Okay, so we'll talk more about the pacemaker cells. The only reason why they're called the pacemaker cells is because they depolarize the fastest. That's the only reason. Because they depolarize the fastest, then the depolarization wave goes throughout the heart from cell to cell to cell. And it's all because of these channels. They're actually called funny channels. That's their name, funny channels, that create an unstable resting membrane potential. So they depolarize, they reach threshold, and they can basically contract all on their own. What happens when something goes wrong with the SA node? That's called the sinoatrial node, right? What would happen? What usually happens is patients, they actually suffer from a lot of fainting. This is called sick sinus syndrome. Dick Cheney, actually, our old vice president, suffered from this. And our own Medtronic came up, the heart device company, came up with the cure, right? They implant pacemakers now. You can actually implant a pacemaker to overcome this particular disorder. All right, so let's go back to skeletal muscle. Let's just kind of bring it full circle here. Skeletal muscle is neurogenic because contraction depends on the motor neuron, right? You can't get skeletal muscle contraction without a motor neuron. So this means it's neurogenic. Genic, again, is origin. Neuro is referring to the motor neuron. And it's excited by neurotransmitters, acetylcholine, binding to those nicotinic acetylcholine receptors, excitation, and then contraction. Now with neurogenic stimulation, you can have multiple or single twitch innervation sites, right? You can actually have summation and tetanus. Another one that we've already talked about, remember the dihydropyridine receptors and the ryanodine receptors? We've already talked about this. Just remember in cardiac muscle, They're not physically interacting. And the dihydropyridine receptors are a voltage-gated calcium channel. So calcium floods in, and it's calcium that actually activates the ryanodine receptors. Calcium is then released from the internal stores as well. And this is a positive feedback mechanism because these ryanodine receptors are activated by calcium, called calcium-induced calcium release. Different from skeletal muscle, remember, the dihydropyridine receptors are not conducting calcium. They actually are physically interacting with the ryanodine receptors, which opens them and then allows for calcium to flood into the intracellular space. Depolarization-induced calcium release. They're mechanically coupled. So for those of you that are in the lab, you've already looked at slides that kind of show these intercalated discs. These are fibers. This is cardiac muscle again. Fibers are long, and they're connected by these intercalated discs. This is where all the gap junctions are, intercalated discs. Now, I think this is really interesting. Let's take a look at the action potentials and the contraction. Action potentials in skeletal muscle, as we already know, is only about 5 milliseconds in duration. And then, so you get the action potentials first, a little bit of a lag time to allow for calcium to flood into the intracellular space, to bind to troponin, move the tropomyosin, and then the crossbridge cycling. Then you get contraction and relaxation. Take a look at the cardiac muscle action potential, right? It is 200 to 250 milliseconds long, right? It lasts as long as the muscle contraction itself. Why is this important? I'm going to have you talk to your neighbors on this one. Why is this important? Everybody's quiet for a Monday. Why is this important? Yeah. Oh, yeah. Yeah, no worries. I think that I'm going to wait till Wednesday for the top hat questions.
Thank you.
Sure. All right, what do you say? Who has an idea of why this is important? I think this is the key word right here. Refractory period. Why is that important in the heart? What do you think? What does this prevent?
Irregular heartbeat.
Yes,
Like rhythmic.
that's true. I'm looking for something very specific, though. It's going to prevent an arrhythmia. Yes, that is true.
Summation or
Yes, that's exactly right. So remember, this is going to prevent submation and tetanus. The last thing you want your heart to do is cramp up, right? That's the very last thing you want it to do. You don't want it to sum and then reach tetanus. It will cramp up and that would be deadly. I'll just say that. All right. So this long refractory period lasts as long as the contraction itself, preventing submation and tetanus. Very important. We'll learn that this plateau phase is actually due to a calcium influx that keeps it very depolarized so that it doesn't repolarize too soon, allowing for summation and tetanus. So if you need some text, this is a really good chart that's giving you some idea of the difference between cardiac and skeletal muscle. And this is an overview of the differences between skeletal muscle, smooth muscle, single unit and multi-unit, and cardiac muscle. All right, I only have a minute left. I can't get started on the worksheet yet. We will start with, please bring this worksheet on Wednesday. We'll start right away with the worksheet. We're going to be talking about mean arterial pressure, flow, and resistance. Ohm's law. Ohm's law. So don't forget about Ohm's law. We're going to be talking about that right away on Wednesday. I think you'll appreciate it.
Most of the problem sets are not that.
Hey everyone, Magna said that your problem sets are in the back here, graded and in the grade book, so if you want to pick those up, you can do that on your way out. Thank you.
These two people, I only got like the first 4 questions.
Okay.
So I'm not sure if like they haven't turned in the rest of it or they...
Maybe, go ahead and like we said before, just they get the 6 for it. So you can do that and if we find an additional sheet, then we'll update it. Yes.
Thank you.
I'm going to give you these two. Magna, I'm going to give you these two.
I didn't come in.
Yes.
I didn't come in Friday, and I sent you an email with the photos
Okay.
of this. And this is actual.
Okay, thank you. Yeah, no.
Are these like...
No. Yeah. Just, yeah, go ahead and just put those in.
I sent you an email last night. It was kind of late. So
Oh, I
didn't
haven't
expect you
checked any of my emails.
Yeah,
yet.
no, that's
Hold
totally
on, let
fine.
me just stop.